We have our first speaker, Tony Wyss-Coray, which we have tried to get to ARDD many times, and every time he says yes to begin with, and then because very enthusiastically says yes, but then something comes up, and because he's an extremely busy man, he's, you know, a star. So thank you so much, Tony, for being here finally, and please go ahead whenever you're ready.
Thank you so much. Thank you, Daniela, Morten, and Alex for organizing this really trend-setting meeting and for having me. I have a couple of disclosures. So the blood is really a tissue that connects all the organs in our bodies, and it permeates these organs. And that leaves the possibility that the blood actually collects molecular information from every organ and from every cell type in these organs. And of course, this is somewhat trivial and has been used for decades in clinical practice, where we can measure molecular markers of tissue function to detect and monitor diseases. But this is usually, again, on diseases, focused on disease, and it's individual single markers that are being measured. So can we use a more broad approach and profile the blood with measuring thousands of different molecules to uncover basic biology of aging and disease? And in this way, study the role of genes, of age, of environment and lifestyle, and learn about biology, but maybe also make models that predict disease for people. We started to do this maybe almost 20 years ago now, and as the assays to measure, for example, proteins have increased, the models have become more reliable. And here's just an example of a study we published a few years ago now, where we measured roughly 3,000 proteins with the SomaLogic platform, which uses aptamers to specifically detect thousands of different proteins. And we did this in 4,300 human blood samples from healthy people in a collaboration with Near Barcela and Sophia Millman at Einstein College, and Adam Butterworth for the interval study, and also together with SomaLogic, a cohort they call COVANS. And here's just an overview of the data from this study, where we show normalized data from these 4,300 people on the x-axis, and on the y-axis you see 3,000 protein measurements for each person. In blue, low levels of a protein, in yellow, high levels of a protein. And what you can immediately appreciate is that age starts at the earliest time point we can measure here. Across human lifespan, you have changes that occur, and these are not linear. And you can also appreciate, if you had a sample from a person at any age, you could probably say how old they are roughly. What we also discovered, and which is very exciting and we still don't understand, is if we look at proteins that change at each given age, and we compare people 5 years younger compared to 5 years older at each age, and we sum up the number of proteins at each age, we get these waves of aging, we call them. And the most striking one is at a young age of roughly 35 years of age, where we see several hundred proteins are significantly different in people older than 35 compared to those younger than 35. We get another wave at around 65, and then the most prominent at age 85 or so. And it's exciting that people have, that other studies are coming out which show, for example, chromatin changes in blood cells at a similar age group, or changes in brain connectivity, for example, also around age 35 to 40 years. So we want to understand, of course, what does that mean, and is this potentially predictive of how you age as you get older? But one key question with all these studies is, are the proteomic changes simply a consequence of aging, of cells and tissues, or of disease? Or might they even contribute to aging and disease? And this is where we had the fortune to be able to collaborate with Tom Rando, who used this old model called Parabiosis he talked to you about earlier today, and you're probably all familiar at this point with Parabiosis, but he was able to show that aged stem cell in the muscle can be rejuvenated in old mice if they are exposed to a young circulatory environment. He also found effects in the liver, and together we explored the effects on the brain and could indeed show that the brain benefits at a molecular, cellular, and functional level from exposure to a young circulatory environment. We were also able to show that a young mouse exposed to aged blood shows an accelerated aging phenotype, and we were able to show that you can reproduce many of these features or these findings of parabiosis by simply transferring the liquid fraction called plasma from young mice into old or from old mice into young. And a very beautiful study that was recently published by Vadim Gladyshev and James White used this model to show that the effects are actually long-lasting. They left the mice together for 3 months and then separated them and could show that even after 2 months of separation, there were long-lasting effects, and the mice, the old mice that were paired to young mice, actually even lived longer than old mice paired to old mice, and they could show that multiple genes, multiple tissues and cells experience the rejuvenation if you look at epigenetic changes or epigenetic clocks. Our lab, together with Steve Quake and the Jen Zuckerberg Biohub, looked at the molecular level, at the transcriptome in individual cell types. We looked at roughly 50 cell types from different organs. Again, in this model, we compared it to normal aging, sort of as the basal truth of age-related changes at the single cell level, and looked at the two paradigms, old mice exposed to young, which we call rejuvenation, and young mice exposed to old, which we call accelerated aging. And what we found is that there are that basically every cell type responds to young or aged blood with transcriptional changes, and that these responses are cell type specific. So there's no common signatures or very few common signatures. But those that we see, for example, in the rejuvenation paradigm are consistent with mitochondrial rejuvenation, activation of the mitochondrial electron transport chain genes, and sort of improvement of aging hallmarks that people have previously described. The cell types that were most responsive and showed sort of a reversal of the transcriptome towards a younger phenotype, interestingly, were mesenchymal stromal cells in adipose tissues, hematopoietic stem cells, so suggesting that there is a rejuvenation of the immune system, as well as hepatocytes, and liver, of course, being a major producer of many growth factors and important immune mediators, is very intriguing that these are also highly responsive to a rejuvenation intervention with young blood. But what we also found is you don't just see reversal of aging, but you also see new gene sets that are established that may contribute to better function. So not just reversal of aging, but maybe the system induces gene changes or transcriptional changes and functional consequences that contribute to the rejuvenation at the functional level that people have reported. Now, one of the key questions, of course, what these studies show is that there are indeed factors in the blood that are able to regenerate and rejuvenate tissues or promote aging. So it's not just a consequence of aging, but these factors may actually contribute actively. And so it's very interesting to figure out what are these factors. But as I showed you, because different cells show different responses, it's most likely that we will not find one factor that can do it all. But individual cells, individual organs may need very specific factors or cocktails of factors to benefit from rejuvenation. So what Hamilton O and Jared Rutledge, two graduate students, set out to do is to try to understand better of what are the factors, what is the basic mechanisms of these effects that we see. And so what we used here is almost 5,000 proteins measured with the SomaLogic platform. And we first identify organ-enriched genes with a gene expression map from GTEx. and then mapped these to plasma proteins to putative organ sources, so that we can say this protein most likely comes from the brain, it comes from the liver, it comes from the heart. Now, it's not the perfect way to do it, but it works pretty well. And so what we found is that 15% of the plasma proteins that can be measured with this platform can be assigned to a specific organ or tissue. Now, most proteins are, or many proteins are produced by many cells, but again, 15% can be assigned to one specific organ. And it's maybe not surprising that the brain has most of these proteins, being the most specialized tissue in our bodies. And roughly 200 proteins out of 5,000 are most likely brain-derived. So this is very exciting because many of these proteins actually change with age, roughly 25% on average, and maybe even more change with different diseases. So this allows us to potentially model aging and model disease processes. And this is exactly what we do. So we can make plasma protein-based models to estimate the physiological state and age of organs. And we do this with the conventional linear regression modeling, such as ENET or LASSO, where we know the chronological age, and based on a large population and the measurements of many proteins, we can estimate the relative age of an individual. And the difference between the actual age and the predicted age, what is called the gap, the difference, gives you information of function, as people have shown with methylation clocks. But while most methylation clocks actually measure the epigenetic age of blood cells, at least in humans, what we can do now is, because we have these proteins that we predict are from different tissues, we can make tissue-specific estimators of age and function. And so we can do this for all major organs and tissues, as well as for the organism as a whole. But again, I want to stress that there is a large amount of knowledge that tissues and cells do not age in synchrony. So having one measure is really not going to be useful, and certainly not a measure of the overall age of a person, as I will show you in a minute. We were able to train these models in collaboration with Carlos Cruchaga at WashU, again near Barcelona, Sofia Millman and SomaLogic, in a cohort from an Alzheimer's research center with lots of healthy controls and people with Alzheimer's disease, and then test this in multiple other cohorts that were completely independent. What we can show, as others with DNA methylation clock, is that in a longitudinal cohort where we have 15-year follow-up, we can calculate risk and predict mortality with most of these organ-specific clocks. Intriguingly, the heart, adipose tissue, are the best predictors of mortality. And if we use sort of the conventional model, basically using all the information in the blood, all the proteins, that does about the same or a little bit less well than the heart-specific clock, because heart disease, of course, being a major inducer of mortality. Now, organs have different ages with the same person, and some individuals are extreme organ agers. So when we select 3 individuals here, which by using the conventional approach, so using all the proteins in the blood, or by analogy using the epigenetic age of blood cells, they all have the same age in the gray circles, but I show you an individual who has several tissues a little bit younger. This person has some younger, some older. This person has most tissues older. Now what we do is we make a outlier analysis where we say if a person has 2 standard deviations above the median or 2 standard deviations below, we call them extreme agers or extremely young. Now, I have to tell you, find very few people who are extremely young in any organ or tissue. But we find 1% of extreme organ agers for almost every organ. Sorry. And these examples are shown here. So this is a person who has an old kidney, but the other tissues are relatively normal age, predicted on their actual chronological age. This person is a heart ager. And then we see a small number, roughly 1% of the population of these people, most of them above age 50, who we call multi-organ agers. And when we use sort of a principal component analysis in a Tisney plot representation, and we just take the extreme ages here and plot the extreme ages of roughly 3,800 healthy people in our cohort, age 20 to 100, we can appreciate this, that there are people who are, have extremely older immune systems, adipose tissue, intestine, liver, pancreas. And this is really very surprising to us. So it's not that you find people who have multiple tissues, let's say the heart, the liver, and the kidney older, but most of them have just one tissue older. So 23% of the people we've analyzed have one or more extreme age gaps, but again, most of them have just one tissue. And 1.2% of them are multi-organ ages. And this is just another way to look at this, a graphical representation. So here we project the relative age of the tissue. 1.56% of the people we analyzed are kidney agers, 1.85% are heart agers, and then 1.19% are these multi-organ agers. And what's really very exciting is that the multi-organ agers are 6.6 times more likely to die within 50 years than people without extreme organ age. This is a huge hazard ratio. And of course, we would want to know, we would want to intervene in these people and identify very early on. But likewise, you would like to find people who are organ ager for a kidney, for a heart, and apply specific interventions. And I just show you a couple of examples. So here's the heart aging model. Uses about 8 or 9 proteins. Interestingly, the algorithm just picks these proteins, and it picked 2 that are used in the clinic, natriuretic peptide and troponin, as heart agers, but we also find new proteins which may lead to additional therapeutic target or an understanding of the biology of heart aging. And here again, in a 15-year follow-up, people who are in the top quartile, who have older hearts, are here 2.86 times more likely to die, to have heart failure or atrial fibrillation compared to the bottom people with younger hearts. And likewise in the brain, we see that people who are predicted to have older brains in the top quartile are here 3.76 times more likely to show cognitive decline over 5 years. This is from an Alzheimer's research study compared to the bottom quartile who are predicted to have younger brains. And this is just a graphic representation of this again. And we find that most people with Alzheimer's disease have older brains. We also find other associations of organ age gaps with disease. For example, people who have older muscle tend to have gait problems. People with older kidneys have multiple metabolic disorders, such as they're obese or they have diabetes and so forth. And again, you can project that on a body map, and you see that Alzheimer's patients have older brains. People with hypertension tend to have older kidneys. People with ischemic heart disease have older hearts. So this is just the beginning. It's still small numbers. We have roughly 6,500 people in this study. We'd like to do it in 100,000 people, and then hopefully we can look at your risk in 5 years, in 10 years, in 15 years if you're an extreme organ ager. So in summary, we find the plasma protein composition changes nonlinearly across lifespan. Cell types respond with specific molecular signatures to changes in blood plasma. These can mimic age acceleration or rejuvenation. And plasma proteomics provides insight into global aging patterns, relative age and physiological state of cells and organs. And lastly, plasma proteomics can identify these extreme organ ages which are susceptible to organ-specific disease. that has implications also for brain diseases. And with that, I'd like to thank collaborators, my lab, and then the funding sources. Thank you.
Beautiful, beautiful work. We have a lot of hands. Rafa was the first, very hard to miss. He was, we need a microphone.
That was fantastic, Tony. Have you thought about having, for example, a model senescence where you target the kidney and do the measurement in plasma pre and post treatment with senescence to see if you are inducing a disease that is known to be age-related, and then you try to resolve it? Would it come back to normal?
With an intervention, you mean?
For example, yes.
Yeah, so we'd love to get our hands on samples of the clinical trials that we heard this morning, for example, from Andrea Meyer, from others, to look at exactly, how does a person respond if they go on to, an anti-diabetic drug, or do we actually change the age? We don't know that yet. We have some retrospective analysis. We see also people who take certain supplements. They tend to have some younger tissues, but not others. So I think it's going to be very interesting. Buddy.
Yeah, hi, Tony. Very incredible, very, very nice. But I have a question about parabiosis. Can you estimate how much the effect of plasma could account for the effect of parabiosis, considering that in addition to factors in the plasma, there's access to younger organs, there's a damage dilution, so presumably it would be only a subset of the parabiotic effect.
Yeah, that's a good point. So we have not done a direct comparison, in part because it's so easy to criticize. Whatever you do, you can't really match the models exactly. As you know, the parabiosis model is a major stress, and especially if you then separate them again, they undergo major stresses, and you could argue that the benefit of young blood is really a recovery from that stress, and it's not a true rejuvenation. On the other hand, with the plasma administration, you have the problem of you do only individual injections. You also have to anesthetize the animals, so you have to overcome that problem. So none of them are perfect, and so it's very hard to compare them side by side. But we do know that plasma administration can mimic many of the transcriptional changes that we see. For example, we just published in the brain, we see very beautiful changes. If we looked at 15 different brain regions, the neurogenic niche is one of the most responsive, and we saw the exact same changes that Anne has reported in her analysis of regeneration and aging of the niche.
Thank you so much, Tony. That was really a fantastic talk.
Thank you.